Hello friends, welcome to CEC Live Lectures. Dear friends, today in this session we are going to talk on photosynthesis. We are going to discuss on the history and concept of photosynthesis. And for the discussion on the topic, we have with us in our studios Dr. Eklavya Chauhan. Dr. Eklavya Chauhan is formerly Associate Professor with Department of Botany, Deshbandhu College, University of Delhi. Dear friends, Dr. Eklavya Chauhan is a prolific professor. He has given 42 years to the academics, and through him we believe that today we are going to get in-depth knowledge on the topic that is photosynthesis. Now I would like to welcome our guest Dr. Eklavya Chauhan once again, and would request him to explain us in detail the history and concept of photosynthesis. Hello sir, welcome to lecture.
Thank you, Geetika. It's a pleasure to introduce the term photosynthesis to students, especially the undergraduate students. This is important because in today's world we have all sorts of problems and very acute problems like global warming, environmental pollution, energy crisis, oil and petroleum shortage. Have you ever realized that all of these problems which I have just enumerated perhaps have one least common factor, and that is the plants themselves, because it is the green plants that are autotrophs, and they are rightly called as the lungs of our planet. If we ask a child as to what is photosynthesis, a good child would immediately answer that plants purify air. That means they take up carbon dioxide and they release oxygen. In fact, this is a term which is an oversimplification. Let us now go into the details of the whole process. I would like to divide the whole chapter, because it's a very, very large chapter, into several subtopics, which would mean that we will begin with history and concept. This would mean that we have first to crystallize the ideas as to how did the whole concept of photosynthesis come into being, how did the equation take so many evolutionary turns during the course of time, and finally what crystallized was a scientifically just and experimentally proven equation. After we have finished that, we will then talk about the nature of light, that is the electromagnetic spectrum, and the wonder pigment which nature has bestowed upon us, and that is chlorophyll. Having said that, the mechanism of photosynthesis assumes unparalleled importance. It is divided into light reaction and the dark reactions, wherein we have photosystems, photophosphorylation, and of course the well-known Benson-Calvin cycle. Apart from it, there are other specializations like the C4 syndrome and the crassulaceous acid metabolism. Not to mention, there is a so-called villain of photosynthesis, which is photorespiration. It has been always seen with a jaundiced eye as being a wasteful process. At the right time, perhaps we would like to justify that after all, it's not that bad as we paint it to be. There has to be something good in nature so as to sustain itself over a long period of time and to have withstood the evolutionary pressures. And finally, we will then dwell upon the factors which are affecting photosynthesis, and more importantly the productivity, because anything which is concerned with green plants has to take into account the productivity. Okay, let us then begin with what are the processes which are taking place in a plant. If we were to see the different processes which are taking place in a plant, basically during daytime it is the process of photosynthesis, where CO2 is taken in the presence of sunlight by the plants, and in process oxygen is added to the atmosphere. Now this particular process is not taking place in isolation. There are other parallel processes which are involving some sort of gaseous exchange. Although carbohydrates are being formed in photosynthesis, they are simultaneously being broken down by the process of respiration. Needless to say, respiration is a process which is taking place night and day throughout, and it is the most vital process. It would take up oxygen from the atmosphere and in turn would release carbon dioxide. Now here comes a balance between the two processes. We know that photosynthesis any day is much faster than the process of respiration, say to the tune of about 18 to 20 times in peak hours. This is very important to maintain the O2 and CO2 ratio on our planet, because if it was not so, then how would we respire? How would the aerobic life survive? In addition, the water vapor is also added to the atmosphere during daytime by the process of transpiration, which also is listed as a necessary evil. Likewise, the veins of the leaf themselves are engaged in conducting water as well as different minerals and of course the sugars which they manufacture to various parts of the plant for their survival. Apart from the fact that all these processes are taking place simultaneously, one should not forget where is the energy coming from for these processes. One would immediately say it is respiration which is burning the carbohydrates. Then the logical question which comes next is, where are the carbohydrates coming from? Obviously, they are coming from the process of photosynthesis. Again, it is basically the autotrophy, which is the most vital process to all survival of life on this planet. Let us begin with a review of the history of photosynthesis. I should say in the beginning that this particular concept of photosynthesis was not made in a day. Like there is a common saying that Rome was not built in a day. It took centuries of ideas to get accumulated and linked in the right sequence and logically crystallized. Then only we had what we call as a final equation for photosynthesis. It was Aristotle, the famous Greek philosopher, who is also considered to be the father of biology and of course the father of zoology. He was the first person to have indicated that plants, like animals, they also require food. Needless to say, there were other saints who were there who had said that all flesh is grass. This was about even 3 centuries earlier than Aristotle. But then perhaps their role was to give importance to plants over the planet. We will discuss about that at the appropriate time. So that means the idea of food requirement had come into the mind of Aristotle, who passed on his information to his student, another famous Greek philosopher, Theophrastus. Incidentally, Theophrastus is also known as the father of botany. In about 350 BC, Theophrastus tried to elaborate upon his teacher's dictum. He said that, okay, if plants are obtaining their food, from where are they getting their food? So he was of the opinion that this food and nourishment was perhaps coming by the roots. So that means the concept of absorption, the concept of nutrition had come into the human mind as early as in 350 BC. One should appreciate the fact that the crystallization of the ideas of photosynthesis need not always have come by the minds of scientists. There were other thinkers also. For example, a saint by the name Nicholas of Cusa, he was in fact a German philosopher and an astronomer. He is considered to be the first modern thinker as far as photosynthesis is concerned. He was a cardinal in his own right in a church. He was the person who tried to give a theoretical idea as to how to prove that the nutrition is coming from roots. His basic predisposition to the idea was that it is water which the plants are absorbing from the roots, but then how to demonstrate how much of water would have given rise to how much of the living bulk today that we call as the biomass of a plant. So he proposed an experiment. It was a purely theoretical idea, not backed by any experiment whatsoever. But it was a very logical one. It was a genius experiment. He said, why don't you pre-weigh some soil in a pot, irrigate it with water, that also should have been weighed, put a plant, and then you, after some time, say a few years, compare the initial and the final weight of the plant, see how much it has grown after a set time. This would definitely demonstrate how much of water has gone into the plant in order to make it grow. This idea was taken up, imagine, after 2 centuries by none other than a Dutch physician and a chemist, Jean-Baptist Helmont. His experiments are the most important, and they are the path-breaking experiments. He took a cue from de Cusa's ideas and he actually performed these experiments. He grew a sapling of willow, which is the Salix species. It weighed about 2.27 kilograms, which is around 5 pounds. He also weighed the clay pot, which was containing soil, so that means he was weighing the soil also, which turned out to be about 200 pounds. He watered this sapling for 5 years, and in 5 years that sapling became a large tree. He removed after 5 years the entire soil, he brushed it, and then he weighed the soil and the plant separately. To his surprise, he found that the tree had increased by 74.39 kilograms in 5 years, which is a little over 164 pounds, whereas the soil had decreased by only 56 odd grams, which is around 2 ounces. Now this was a very perplexing situation, because how could such a large bulk of biomass be formed by such minimum of soil? So the answer was very clear in his mind. He concluded that the entire bulk of about 164 odd pounds of wood, bark and roots, which was the biomass, was formed by water alone, and he did not give an iota of importance to soil or its minerals, because there was no concept at that time. So his results were published after his death in his classical book on the power of medicine. In today's perspective, we would say that he was partly correct only. Why? Because there was no idea of gases from the atmosphere. There was no concept as to whether sunlight would also be playing a role, and of course nothing about mineral nutrition. So it was a half concluded program. However, it was a path-breaking one. At least an experiment had been done. It was John Woodward who for the first time talked about, definitely about, the nutrients which were being taken up from soil for the growth of a plant. Incidentally, John Woodward is considered to be the pioneer for the study of hydroponics, which he developed in 1699. And today hydroponics is so very popular. You can control the growth of a plant for fruits and for vegetables by manipulating the mineral content of soil and all the other benefits of an aquaculture or soilless culture. He took the mint plants or the mentha plants and he grew them in water of different purity. He took distilled water, he took tap water, river water and muddy water. It was such a wonderful experiment, such a simple and elegant experiment, and he found that the muddy water was the best for the growth of a plant, thereby concluding that there has to be some matter in mud which is dissolved in water and it is being taken up by the roots, and this is how it is providing nutrition to the plant. This led to the famous Humours Theory, which means the humus in soil holds the key to the nutrition of a plant, and this is how growth would be taking. Water is good because people thought that water is the most important one, and Woodward said water is important, but then it is a medium for transportation and the rest is done by the humus. Then came to the scene the British Stephen Hales. Incidentally, Stephen Hales was the person who gave the term root pressure, and he is also considered as the father of plant physiology. He had his very famous book Vegetable Statics in the year 1712, wherein he made a very bold statement. He said very probably the plants or the leaves, they derive their nourishment from air, and this process may involve light. So that means we have now 4 ingredients: water, soil, air and now light. Nobody before Stephen Hales had given any importance to air or sunlight, and this guess of Stephen Hales was finally proven right by the subsequent scientists. There was a Swiss naturalist by the name Charles Bonnet who observed, incidentally, emission of gas bubbles from the leaves of illuminated submerged hydrophytes. Now, any experiment that you see in your school or colleges today for photosynthesis, it is the simplest and the most elegant experiment wherein the hydrilla sprigs are taken in an inverted funnel and oxygen gas collected. This is the easiest way to demonstrate the process of photosynthesis. It was observed as early as in 1754, not knowing what was going on, what was taken in, what was being produced, what was the whole purpose of the process, and the term photosynthesis was not there at all. We will talk about it shortly thereafter. The English clergyman and chemist Joseph Priestley was a very, very prominent scientist who at time of his work, which is in the 1772, he was a person who was working on the nature of different types of gases. At the end of the 18th century, there were people had a lot of notion about impure air and pure air, because anything which was being burnt was producing impure air, which they called as phlogiston. Phlogiston is some sort of foul air which could be released by burning of flame or any such activity. They were all bothered as to how is this impure air to be purified. In other words, how is this phlogiston to be converted into dephlogiston. This was the basic idea. At least Priestley was able to talk about impure air and pure air, and he performed a very elegant experiment. He took a set of bell jars. In one bell jar, he took a burning candle and he left a mouse inside to play. After some time, he observed that the flame went off, and so did the life from the mouse. They were not able to survive in this environment. So this proved that there was something going on, and some foul air or phlogiston was being produced, which did not allow the mouse to survive. In a second set of experiment, he took again a burning candle along with a live mouse, but he also introduced a sprig of mint, that is mentha, and to his surprise he found that the candle went on burning, and the mouse was playing as usual. So that means he concluded that the animals, they are producing some sort of foul air, so the candle cannot burn in this foul air, and when the plant was introduced, it restored or it dephlogistated the foul air. So that means the candle would burn or the mouse would have remained alive only when the air was pure. This was a very important experiment, but then it had some shortcomings. The parameters which the earlier scientists or the Joseph Priestley had missed was perhaps filled in by the next scientist, Jan Ingenhousz, who was a Dutch physiologist and a chemist. He said that the work of Joseph Priestley is perfect, but then what he perhaps forgotten was that this purification of air by the plants would have been possible only in the presence of sunlight, number one, and number two, it would have been possible only if the plant which was put inside the jar was green. So that means two parameters were now being established gradually, that is the role of sunlight and something to do with the green color of the plants. When we say green, that means if you had kept a mushroom, perhaps things would not have worked the way Priestley wanted it to work. So that means non-green plants would not do this job of purification. So indirectly, Ingenhousz had recognized the participation of chlorophyll, which was discovered much later, and of course the role of sunlight, which was proved indirectly by his experiments. So one thing which also came to the fore was that plants simply are not purifying the foul air alone, but what is happening at night? Since there is no light, the plants themselves are generating bad air, which is phlogiston, and this is perhaps the reason why he even recommended that the plants should not be kept indoors at night, lest they cause toxicity to the occupants. I think this is an advice which is followed even today, and it's a matter of common sense till we reach the camp plants. The matter was sorted out to a certain extent by none other than Anton Lavoisier, who is also hailed as the father of modern chemistry, because till now people were talking about foul air, impure air, pure air, but nobody knew what was the chemical composition of this air. So he was the person who analyzed the pure and impure air, and he came to a conclusion that what people were talking about as phlogiston was nothing but oxygen. And this terminology came in the year 1778. Although the scientist by the name Joseph Black had already discovered carbon dioxide. So that means when this discovery was made, then the phlogiston theory was disproved, because nobody knew earlier that plants also were generating foul air at night, and they were sort of purifying it only during daytime. So in fact, Ingenhouse reinterpreted his results in the light of Lavoisier's work. He said, okay, if you are saying that carbon dioxide is being released, so the plants, let me just put it again in the words of Ingenhouse, he said that plants will split carbon dioxide in the presence of sunlight, and carbon of this carbon dioxide would now be utilized by the plant for its growth. In the whole process, oxygen is being given off as waste. Now, this modification itself is not entirely correct, because as we would see subsequently, it's not the carbon which, or the carbon dioxide which is the source of oxygen, but this would take maybe another 50 years to remember. So this had to be modified. The whole concept, it was really destined to be modified 1.5 centuries later by none other than a graduate student himself, Cornelius B. Van Deel. This is how the whole concept came. Another scientist by the name Jean Senebier was the first person, and he was also obsessed with the idea that carbon dioxide is essential for plant growth. He said, let me just prove it. He boiled some water, so there would not have been any carbon dioxide in it, and then when he put a submerged plant, there was no oxygen being produced. So that means he concluded that oxygen is definitely coming from carbon dioxide. Surprisingly, Ingenhouse also had agreed with Senebier. So till now, the theory was that carbon dioxide was being split and oxygen was being produced. In other words, green plants were nothing but they were like an air purifier in the semantic sense. It was like a machine which was taking up carbon dioxide and releasing oxygen. In our second part, perhaps we would dwell upon more on as to whether this whole idea was correct or it needed a big surgery.
With this note, thank you, sir. Thank you so very much for giving us a vivacious session. Dear friends, you're requested to be with us as we are back after a short break and would be discussing more on photosynthesis. Thank you. Hello friends, welcome back to this session. Friends, as you know that today we are talking on photosynthesis, and specifically we are covering on the history and concept of photosynthesis. For the discussion on the topic, we have with us in our studios Dr. Eklavya Chauhan. Dr. Eklavya Chauhan is a renowned professor of botany, and through him in our previous past also we discussed on various issues and topics and gathered lots of information. We believe that through this session also you are going to grab maximum. Now I would like to welcome our guest Dr. Eklavya Chauhan once again. Hello sir. Welcome.
Thank you. As in the previous session, we were talking about the obsession of the scientist Jean Senebier. The entire episode highlights how the wrong concepts run for centuries, even in the minds of scientists. This is primarily because of lack of direct experimentation or a mindset. So that means by this time of Jean Senebier, that is by 1796, people were convinced that oxygen which is being released by the plants is coming from carbon dioxide. And this is how the entire story proceeded. And to give it a scientific credibility, none other than the famous scientist, a Genevian of French descent, Theodore de Saussure. He even made quantitative gas measurements. His results may be true even today, but then the interpretation is entirely different. He found out that the release of oxygen was equal to the consumption of carbon dioxide. Fine. But then he also gave an idea that the organic matter which was produced plus oxygen, this was much greater in value than the carbon dioxide fixed. So that means, how could carbon dioxide alone account for the formation of the entire biomass? So in other words, the water is also important, and as a result an equation emerges, and that is carbon dioxide plus water plus light would give rise to organic matter and oxygen. We all agree there is an equivalence between carbon dioxide and oxygen, which doesn't necessarily prove, this equivalence doesn't necessarily prove that CO2 is the source of oxygen and nothing else. In the meantime, two very important discoveries were made. Two Frenchmen, Pelletier and Kevin-Tau, they were responsible for isolating a green pigment from the leaves, and this was termed chlorophyll. This wonder pigment had no role in photosynthesis till another Frenchman, Dutrochet, in 1837 established that chlorophyll is absolutely essential for the plants to assimilate carbon dioxide, and the yellow leaves are in no position to make any food whatsoever. The a scientist by the name Hugo Von Mahal made a very important discovery, because he was the first person who described as to where these chlorophyll molecules are located. He had given a German term, chlorophyllkornern. That means the chlorophylls are present in the form of small granules, which were later termed as plastids, and finally they were christened as chloroplasts in the year 1883. One should not forget, Hugo von Mahal was the person who is known for discovering protoplasm in plants. The work of another German physicist and a physician, Julius Von Meyer, is very important. And one would like to read a paragraph of his original saying, which I believe sums up many things. He said that energy is neither created nor destroyed. Now this was for the first time that the law of conservation of energy came into the minds. Till now we were talking about carbon dioxide, oxygen, water, some organic matter, but then what is the basic purpose of a plant or of a green leaf? What is it doing? So that means he made his claim in his book entitled Organic Motion in Relation to Metabolism, and this was a masterpiece. He said that nature has to catch something from the sunlight, and he was the one who really underlined the importance of the process. In his words, nature has put itself the problem how to catch in flight the light streaming to the earth and to store the most elusive of all powers in rigid form. And to achieve this aim, it has covered the crust of earth with organisms which in their life processes absorb the light of the sun and use this power to produce a continuously accumulating chemical difference. So that means it is these converters, they are nothing but they are these organisms or plants. So the plant kingdom forms a reservoir in which the fleeting sun rays are fixed and so very skillfully stored for future use, an economic provision to which the physical existence of mankind is inexorably bound. That means the plants take one form of power, light, and produce another form of power, which is the chemical difference. This was the first time that somebody had used the terms power for sunlight and chemical difference for chemical energy. To put it in simple words, physical energy was being conserved as chemical energy. So this changed the whole idea of our thinking. That means plants produced organic matter as well as provided energy. In other words, plants were not merely purifiers, they were converters of one form of energy into another. This led to another very convincing equation, and that was carbon dioxide plus water in the presence of light was releasing oxygen, some organic matter and chemical energy. This of course is not a balanced equation. This reminds us of the biblical saint Isaiah, who 700 to 600 BC had given his prophecy that all flesh is grass. It is such an old concept of giving reverence to plants that the entire life on this planet depends upon the activities of plants. What else if not photosynthesis, minus of course the genesis of the term so far. Then came the French chemist Jean-Baptiste Boussingault. He was a person who made the scientific approach of carbon dioxide and oxygen in the form of quantitative measurements, and he gave the idea of assimilatory coefficient, which means oxygen and carbon dioxide in the ratio was always 1. His intentions were very good, but then the formulation of the equation again strengthened and confirmed our ideas, which later we would find to be a misconception, of equivalence being the source. Equivalence it is, but source it is not. That means 6 molecules of carbon dioxide giving rise to 6 molecules of oxygen, and of course the formation of some sugars in the presence of sunlight and chlorophyll. And the glucose theory was strengthened by the German physiologist Julius von Sachs, who made one of the first physico-chemical studies on photosynthesis, and he was the person to prove that sunlight results in the synthesis of starch grains in the leaves, and in dark, if we analyze the leaves, the starch grains are gone. So he was the person who for the first time established that the most abundant storage product of photosynthesis or of this assimilation is starch, of this entire process is starch. The classical experiments of Engelmann, who was a German botanist and physiologist, for the first time suggested to us that chlorophyll is not functioning in isolation, it has a relationship with light quality. Agreed that the chloroplasts are the sites of photosynthesis, but then the chlorophyll has different roles to play in different lights. So Engelmann performed 3 classical and very ingenious sets of experiments, and which helped us to establish many concepts. He was the first person to have used spectroscopic techniques, by which he came to a conclusion that all wavelengths are not equally absorbed by chlorophyll, and likewise all the wavelengths do not induce equal rate of photosynthesis. And on the basis of his premises, he gave the concept of absorption spectrum and action spectrum of chlorophyll. He said that if a graph which is plotting light absorption versus of the chlorophyll versus wavelength, then we can find out which is the best wavelength of light which is being absorbed by chlorophyll. And likewise, if we plot a graph between the rate of photosynthesis and the wavelength that we are providing, we can also find out as to which is the most effective light part which will propel the intensity of photosynthesis. To understand this concept, let us view the electromagnetic spectrum. Out of the entire gambit of rays which are starting from sun and falling on earth, one should appreciate our eyes are sensitive to only just a very narrow range of wavelength, and which is the Vibgyor or the visible range, which is ranging from 400 nanometers to 700 nanometers, a very, very narrow range of the entire spectrum. And on the left side is the ultraviolet, the X-rays, the gamma rays, which we can perceive but we cannot see. Likewise, on the right side, the infrared, the microwave and the radio waves. They are also not, they can just be felt but not seen. So that means as the spectrum goes from the left to the right, there is a decrease in energy, there is an increase in wavelength, and both these terms, they are inversely related. So one can see only the visible spectrum only between 400, which is the violet, to the red range. The absorption spectra of chlorophyll, which we can see in the modern terminology, and we will dwell upon this more in some of the next lectures, or if one sees clearly, chlorophyll A has an absorption peak somewhere 435 to 440 nanometers, and then there is a sudden drop and there is a sleeve. Now this area is the green region. Of course, how can chlorophyll act in green? It would it will not absorb anything. Green does not absorb green, it reflects. And likewise, the second peak of chlorophyll A is somewhere at 662, which is the red region. So that means this proves a point that chlorophyll absorbs light in the violet blue and in the red region, and this would be the absorption spectrum. If we compare this to the action spectrum as to what is the photosynthetic rate versus wavelength, you would see that there is a remarkable collinearity. That is, if this is the absorption, this is the action, nearly, we would not say entirely overlapping, but nearly overlapping. Likewise, in the red region, this is the activity of chlorophyll. Now this, no wonder, this is not a strange coincidence, but it is a coincidence which is running the whole show of life on earth, because this is giving maximum efficiency to chlorophyll for photosynthesis. In other words, it was not a fluke, it was a definite, predestined process of nature which led to the evolution of this pigment as the principal photosynthetic pigment. Not necessarily right from the beginning and not necessarily in the future, but then right now this is, and a few geological years, this would be the main photosynthetic pigment. Engelmann performed some very simple but elegant experiments. He worked on the filamentous green alga Spirogyra. A special microscope was fitted, it was taken from Carl Zeiss, and a special type of prism was fitted, and this could produce a spectrum on the microscopic slide. So that means on the slide itself one could produce 3 different wavelengths coming out from sunlight, and whether to find out which was the most effective wavelength, he took a drop of aerobic motile bacteria, which we call as aerotactic bacteria. They could they are attracted to oxygen, and they would accumulate only in areas where oxygen was rich. So he put a drop of bacterial suspension on the slide, and the results were very fascinating. These two are similar results, but they convey the same idea. The Spirogyra is the filament which was put on the slide. A prism was taken which was now dividing or compartmentalizing light into 7 different wavelengths, and when it fell on the different regions, it was found that the aerotactic bacteria got accumulated at 2 regions. One region was blue and violet, and the so that means you can see the green and yellow, they were all left, and indigo, it was red. So that means these 2 areas which were illuminated with blue and red light attracted most of the areas. This suggested that these 2 are the most effective wavelengths. And if we compare this with the spectrum, you can see it directly matches with the spectrum. So Engelmann was the first person to give an idea as to blue and red wavelengths are the most effectively absorbed ones. Surprisingly, there are purple bacteria also. They do not take visible spectrum of light. They are photosynthesizing best when they are given UV light. And UV light we cannot see, we can the bacteria can function the best. So that means, in a nutshell, we started in early 17th century and we arrived here by this position in 1864. Imagine, still no scientific term had been given to the process that we have been discussing so far. However, the equation was made of the equivalents: 6 carbon dioxide, 6 water, light energy, chlorophyll, giving rise to a molecule of a hexose sugar, and 6 molecules of oxygen, but no term at all. The christening of the term, or giving the name to a process, was the regime of Barnes, who was an American botanist. Till 1893, people were talking about this process as assimilation of carbon or simply assimilation, but no formal term was given, which was universally acknowledged. So he proposed 2 terms, photosyntax and photosynthesis. And he even defined photosyntax. He said photosyntax is some sort of synthesis of complex carbon compounds out of carbonic acid, which means carbon dioxide, in the presence of chlorophyll under the action of light. Personally, Barnes liked the term photosyntax. His results were discussed at a meeting of the American Association of Advancement of Science in Wisconsin, USA in 1893, where his friend and another fellow American botanist, Conway Macmillan, had his own say. He advocated for the term photosynthesis. He said both the terms are good, but photosynthesis is a more expressive term, and from now onwards that means another 30 years where before the term photosynthesis actually became acceptable. So just to give you a quick rejoinder, it took almost 2000 years when Priestley confirmed Aristotle ideas that plants required food. So it took about 20 centuries, and it took almost 1 century again after Priestley. That is when Bossingau's work in 1864 resulted in the equation for photosynthesis, and after the equation was given, it took another 30 years before the photosynthesis term became universally acceptable. Then, ushering into the 20th century, we know that things moved very quickly now at a very fast pace. There was evidence for light and dark reactions. Scientists like Blackman, Warburg, Emerson and Arnold, many of them Nobel laureates, they also helped to establish the presence of two types or two parts of photosynthesis, namely the photochemical or the light reaction and the thermochemical or the dark reaction. However, even in the beginning of 20th century, the whole concept of carbon dioxide and oxygen equivalence was so very firmly rooted in our minds that we were not able to get out from this, and everyone believed that oxygen came from carbon dioxide. Now the $1,000,000 question arises, is it really true? The whole concept was made topsy-turvy in the year 1924. As I mentioned earlier in part 1, the American microbiologist Cornelius Von Meyer, who was just a graduate student, he was given a problem by his professor to find out whether any green sulfur bacteria also photosynthesize and give water. Just try to find out. So in his thesis he found that there were certain green sulfur bacteria who had a slightly different type of chlorophyll, which was bacteriochlorophyll. And to his surprise he found that these particular bacteria were not utilizing water at all. And his surprise became greater when he found that they were not releasing oxygen at all. Instead, they were releasing elemental sulfur. Now, in the year 1924, it became clear that even photosynthesis is of two types. First, oxygenic, where oxygen is being produced, and anoxygenic, which is without the evolution of oxygen. And throughout 300 years, we have been advocating that oxygen is being released from carbon dioxide. Here, there is no oxygen being released at all. So how does one place oneself? So Von Meyer's idea was that oxygen comes from water. Why? Because he said that water would be photolysed, and this idea was absolutely contrary to Jean Senebier, who had started the whole concept that carbon dioxide is being split and oxygen is being released. So Von Meyer said H2S is giving rise to protons and electrons and elemental sulfur. Likewise, water also must be splitting, and it would be giving rise to protons and electrons and half a molecule of oxygen. In other words, Von Meyer was the first person to have perhaps indicated that photosynthesis is also some type of a redox process, wherein reactions of reduction and oxidation are taking place. So at the end of his thesis, very reluctantly, C.B. Von Meyer gave an equation, because this equation had to be a revolutionary equation. It had to break all the earlier myths of the carbon dioxide being a source of oxygen. So he said that if my equation, namely 6 molecules of carbon dioxide and 12 molecules of hydrogen sulfide in the presence of light and bacteriochlorophyll can synthesize a hexose sugar, and 6 molecules of water and 12 molecules of sulfur, of elemental sulfur, why not consider an analogous situation wherein what we call as the oxygenic photosynthesis also behaves in a similar manner. In other words, it also releases, it takes 6 molecules of carbon dioxide, and instead of 6 molecules of other things, it takes 12 molecules of water, releasing 6 molecules of water and 6 O2. Now this equation nobody accepted, and they said that this equation may not be that well. However, the subsequent work which was triggered by this departure of C.B. Von Meyer, it went dormant for many years, and it was much later that the scientists actually proved that how correct the young graduate C.B. Von Meyer was. I think it will take another session with us to really come back to that revolutionary idea, which crystallized finally into giving a more authentic, empirically more correct, and physiologically more sane equation for photosynthesis. Thank you.
With this note, thank you, sir. Thank you so very much for giving us this session. Friends, you are requested to write to us at info.cec@nic.in. Here you can post your questions as well as your feedback too. We are going to meet again very soon and would be discussing more on photosynthesis. Till then, take care. Goodbye. Thank you, sir. Thank you once again.